present my research title. My research title is on application of magnetic separation technique in various biological processes. So far you have heard mainly the magnetic field, but it is a little bit different from that field. It is in combination with the magnetic science and biology. So, what is magnetic separation? Magnetic separation is the process of separating magnetic substances from the mixture of non-magnetic substances with the help of magnetic powder and magnet. Although magnetic separation is a physical process, but we successfully applied this technique in the wastewater treatment as well as in citric acid fermentation. Therefore, magnetic separation techniques are promising for various biotechnological applications. You know that this sensory is the sensory of biotechnology. So, this magnetic science would help biotechnology very much. Now the textile dye industries, the largest water polluters. Industry is a huge source of water pollutants that are harmful to people as well as the environment. The textile industry is one of the largest polluters in the world. World Bank estimated that about 20% of global industrial wastewater pollution comes from textile dye. In Bangladesh, you may know that, or is Bangladesh? It is an Indian subcontinent. It is once upon a time it was a part of India, then Pakistan, now it is the East Pakistan now became the Bangladesh in 1971. So in Bangladesh, we have many rivers. We call it riverine countries. So rivers are highly polluted by the domestic as well as industrial discharges now. Therefore, we have taken a research program on textile wastewater treatment by magnetic activated sludge. We call it MASK process to address this burning issue. Since we are biologists, we are deep always in the biological system. So biological wastewater treatment, it is eco-friendly as well as cost-effective process. If you compare with the physical and chemical process, so it is always cost-effective and eco-friendly process. The conventional process, we call it activated sludge process. And our method, we call it new process. It is simply modification of the conventional activated sludge process. It has two parts, the conventional mass process for especially the domestic wastewater. The right hand for less biodegradable, or we may call it xenobiotic compounds. We are synthesizing regularly for new compounds to address our problem. So that is we call the advanced mass process. This process we are divided into two parts, in 2 reactor systems and 1 reactor system. I will explain these systems sequentially. Now I would like to explain what is activated sludge. Wastewater contains some suspended solids. We are regularly discharging wastewater after using, and that wastewater contains suspended solids, and when agitated in presence of air, the suspended solids form neatly on which biological life develop and gradually build up to larger solids are known as activated sludge. Activated sludge process was developed in England in 1940, and it involved the production of an activated mass of microorganisms capable of aerobically oxidizing the organic contents of waste. Activated sludge is probably the most versatile biological treatment process capable of measured BOD. One major problem is the excess sludge management generated by wastewater treatment. What's BOD? BOD is a biochemical oxygen demand. You know the water, if it contains the pollutants, organic pollutants, microorganisms need to oxidize that's organic compounds and needs oxygens for their biochemical system. That's why we call biochemical oxygen demand for oxidizing the organic compound present in the wastewater. We generally, organic loads, we measure in the form of either BOD or COD. COD is chemical oxygen demand. So this is the diagram process of the activated process. We call the activated sludge process the wastewater cleaner. So this is the main tank where the microorganisms, we call the activated mass of microorganisms, are working as a laborer. And after consuming the organic compounds present in the wastewater, it needs separation in the sedimentation tank. After sedimentation, the treated water coming out. During this process, since it's a biological system, the new cells are developed. So then we have to need to withdraw excess sludge. About 20% of the organic compound consumed by the microorganisms. That is the big problem to address this dumping the excess sludge. So look, this is the wastewater cleaner. We call the activated sludge aggregates of microorganisms. Look, this is mostly bacteria. But look, this is a protozoa. So various types of microorganisms present with this activated sludge mass to consume the organic compounds present in the wastewater. You can think we are using water for the washing, using washing water for the kitchen, then we polluted the water that comes from our system. So cleaning of wastewater is a messy job, but magnetic powder promises to make the process more efficient. So this is the right hand side. It is the modification of this activated sludge process. Activated sludge. It is simply modified by this is magnetic powder, that is we call iron powder. Why is using this? Because we need magnetic separation of this sludge. By using this magnetic powder, the magnet can grab this sludge. So this is the diagrammatic model of the magnetic sludge process. So this is the magnetic activated sludge. Since it is a modified form, it contains iron powder within the sludge system. Then this is a magnetic drum. It's a very simple drum. It's a plastic barrel covered, acrylamide barrel covered with a plastic magnet, so that it became the magnetic drum. This magnetic drum can attract this magnetic activated sludge. After treatment, this water is coming out from this work. The magnetic sludge attracted by the drum here and separated by the scraper, and it's coming again within the sludge. So, since it is a new process, it has one advantage, especially we call it zero emission of sludge. That in this process, we need not withdraw any excess sludge. And this is the pilot plant. It is running about 10 years in the Utsunomiya city. It is successfully running on. This is the performance of the pilot plant experiment. Look, the COD chrome, that is the organic compound removal, was measured in the form of COD. We are about more than 90% organic compounds removed successfully. Similarly, another pollutants, that is ammonium nitrogen, or more than 90% was removed. So magnetic separation, it is very fast. Look, fraction of a second, that is within 10 seconds, the suspended solids can be separated through this process. But in case of activated sludge, it needs 2 to 3 hours. So you can compare how it is faster, this magnetic separation process. And we call it the zero emission of sludge. This figure we could explain how this is zero emission of sludge. Look, the biomass concentration is remain the same concentration. We are keeping the concentration in such a level, it looks like a suboptimum nutrient supply, so that self-growth and self-decay, keeping the biomass in the balanced condition. That is the merit of the magnetic separation. In brief, this is the comparison of the conventional process. This is the magnetic activated sludge process. Here we use magnetite, sedimentation tank absent, sludge separation process. In case of conventional process, is a gravitational force, but in case of magnetic activated sludge process, it is a magnetic force. The one advantage is, in case of sludge emission, AS has yes, but in case of mass, there is no sludge emission. We call it zero emission. So it is very mass friendly. So this is the diagram of the textile dye wastewater treatment process. We have studied 2 systems. The first one is 2 reactor systems, and second one is the 1 reactor system. Now I would like to explain this way. In the textile dye wastewater, 2-type dye, especially the azo-dye, it is very difficult to break down because of this nitrogen-nitrogen bond, this azo bond. That's why we need 2 mode of operation. One is anaerobic, another one is aerobic. In aerobic